Hi, everybody. I have to say I am really delighted to be here today. I have visited this community once or twice, but I don't know it very well, and I'm really hoping to build closer links between myself and this community, but also between what's happening in Melbourne and around Australia in Huntington's disease research. So this is a time of great hope in Huntington's disease. 2018, it's the best news and the best year for Huntington's disease since 1993, when the gene was first identified. Now, I'm sure all of you know that although we have some treatments that can help to improve the symptoms of Huntington's disease, and we learn more and more about how to care for people, there's a lot we can do for Huntington's disease, but we don't have, at this point, any treatments that can slow the progression of Huntington's disease or delay the onset. But we are hot on the trail, and this is what many people across the world are really working toward. And I think 2018 has really seen a lot of progress, and that's what I want to tell you about today. So how many people saw something in the news or heard a buzz about this at the end of last year? How many people? Oh, look at that. Wow, a well-informed group that you are. So what I want to do today is I want to talk about this news, and I want to break it down and talk about what it means, okay? And then I want to talk about what can be done, in terms of the people here, this community, and what kind of role you might be able to play in helping to forward and further the research efforts that are going to lead us to the treatments that are going to make a difference for slowing the progression of Huntington's disease, delaying the onset, and thereby really affecting the disease itself. Okay, so I want to kind of emphasize there's a lot we can do now, but we want to do much, much more. Just got to wake up the mouse, I'm told. Wake up, mouse. Okay. So I told these guys I'm a bit of a walker, so I have to kind of shift over here and see what my slides say. Okay, so this is what I'm going to do. This is a quote that comes from, it's all over the web, but I just picked one and I put it up here. This one's actually from Italy, okay? So this is Sarah Tabrizi. She's a professor at University College in London. And this is a quote from an article that was highlighting the findings from the IONIS study. It says, "The best news for the Huntington's disease community since the discovery of the gene. IONIS trial data revealed, Roche confirms jump to phase 3. All right, let me translate a bit of that for you because it's kind of obvious what it says, but I want to tell you what the underlying meanings are. So the first thing is IONIS is a small biotechnology company. It's not really small, there's smaller ones. It's in kind of just north of San Diego in California. And what they do is they develop novel technologies that address many different disorders. And they took an interest about 5 years ago in Huntington's disease. And I'm going to tell you a little bit about their technology in a few minutes. But why do we have Roche up here? Well, Roche is a huge pharmaceutical company based in Switzerland, but it's all over the place. So what happens in drug development quite a lot of the time is smaller biotechnology companies start developing drugs. They try novel methods and they do some of the initial studies, but they usually don't have enough money to run these huge trials that you need to run to get drugs approved so that they can be for sale to the public, okay? So when IONIS got some early promising results, and people like the strategy that they're using to come up with ways to treat Huntington's disease, what happens is then Roche came in and made a deal with IONIS that said, if you get promising results in your phase 2 trial, we will license the drug from you, pay a lot of money for it, and we will move it into phase 3 and spend the money to conduct a really large trial. So in December, IONIS had some really promising preliminary results, which I'm going to talk about. And when that happened, Roche swooped in and said, we are here with money and we want to do this work with you and we're going to help to develop the drug. Okay, so that was fabulous news for two reasons. One is because there was a promising result from their kind of more preliminary study. And two is there was going to be money behind the development of this drug to take it into the future. It'll probably cost on the order in this next phase of maybe $50 million or something like that to run the trial. It's a lot of money. This is the next announcement. So this announcement was made, this announcement was the December announcement. So this is what they said precisely. The phase 1/2 2A trial for the gene silencing drug, Ionis HTT-RX, that's the name of the drug. This is what they said, quote unquote, substantially exceeded our expectations in safely reducing the mutant Huntington in the CSF. Okay? Fabulous news. So what does this mean? So phase 1/2A, that's the most preliminary kind of study there is, okay? So it was a very small sample. It's about 46 people is how many people are in that trial. I'm actually involved in the trial because I developed the cognitive assessment component that's being used to look at whether cognition improves with this drug. What it means when they said it substantially exceeded our expectations, I'll tell you what happened there. This drug is actually injected into the CSF, and that's the cerebrospinal fluid. It's the fluid that flows around in your brain and down in your spinal cord, okay? And how that's done, it has to do with a lumbar puncture or a spinal tap, okay? In a lumbar puncture or a spinal tap, you get a little needle inserted, usually at the base of the spine, and you can take fluid out, a little bit of that cerebrospinal fluid, and you can, measure what's in it and that sort of thing. It's also where this drug is injected. So when they do the drug injection, they take a little bit of fluid out, and they put a little bit of fluid in that contains the drug, okay? And when they took the fluid out, they wanted to measure how much Huntington protein was in the fluid. And at the beginning, before people had treatment, they collected the fluid and they saved it. And then after the treatments and the placebo treatments were administered, then they took another sample to compare it. And what they found is that in people who had received the Ionis HTT-RX drug, that there was a reduction in the Huntington's protein found in their CSF. So this is great news, okay? So we're going to talk about that a little bit more. Now that's not enough to get a drug approved so that it can be sold in the market, because no drug gets approved just because it lowers a protein in the CSF. What it has to do is it has to improve clinical symptoms. So at this point we had no evidence about that. We didn't know if that was going to be the case or not. We still don't really know, but we have some early indications, which I'll get to in a minute. Okay, this is the next announcement, okay? So this is March 1st, 2018, at the CHDI Therapeutics meeting in Palm Springs, California. I happened to be there and heard this announcement. This was the first scientific presentation of the data. So up to this point, all we had was a press release. And scientists are not very satisfied with just a press release because we have what's called peer review. Data need to be presented and evaluated by other scientists to help to determine whether the significance of the data warrants the interpretations offered by the pharmaceutical company. So this is the announcement in the press release that came after the CHDI presentation. So this was the first hearing of this result by the scientists. This is what they said. The Ionis HTT-RX lowered mutant Huntington an average of 40% with a maximum reduction of 60%. So that's substantial. This is not 5% or 3%. This is clear evidence that this drug is having an effect, okay? Now, a few important things to say. This doesn't say anything about safety, and the primary goal of the earliest studies is to establish the safety of the drug. Now, it does look like a pretty safe drug, but it needs to be tested in a much larger sample to establish that for sure. And sometimes drugs that look really good at this phase do fail safety because people do get complications and they can't be used. And that's happened in many drug development programs. So we can't be at all sure yet, but we are very excited about the potential of this treatment. And this is kind of the last press release I want to talk about before I go into a little bit more about the mechanism. So this was a press release from April 24th. This was a press release that said, new data from Ionis HTT-RX phase 1/2 study demonstrates correlation between reduction of disease-causing protein and improvements in clinical measures of Huntington's disease. All right? This is looking good. Because what this is telling us is not only the drug showing us a proof of principle that it actually does appear to lower Huntington's disease in the CSF, now we see the first bit of evidence that it is translating to improvements that are meaningful to people. Because I don't think any of you really think it would be that meaningful just to have a protein go down if you didn't get some benefit in terms of the symptoms and signs of the disease. So this tells us that they, in their preliminary study of only 46 people, they begin to see that clinical improvements may be possible with this drug. You know, when this announcement was made in the previous press release announcement, the one that occurred at the scientific meeting, do you know the scientists in the audience, which were about 600 people, they stood up and clapped and cheered. It was fabulous. Okay, so this is that press release that started all of this news for us, and we've been really excited about it. Sarah Tabrizi is a clinical neurologist at University College London, and she's been a key driver from the academic point of view in helping to guide the Roche and the Ionis companies in developing the way this clinical trial is going to run. And she was the leader of the TRACK-HD study, which some people may know something about. The TRACK-HD study is what's called an observational study. It didn't do any interventions like treatments or like drugs or any other kind of treatment. But what it did is it looked really closely at the symptoms of people that people experience with Huntington's disease. It looked at their cognitive symptoms, and in fact, I was in charge of that component as well. So I developed a battery that helped us to characterize how people were doing cognitively. We looked at people who were prior to the disease diagnosis, people who were after the diagnosis, and we compared them to people who didn't have the gene. We also looked at their movements in great detail. We looked at their psychiatric function, the mood disorders, and that sort of thing. And in addition to that, we took a really close look at the brain using MRI. And this data was really essential to the development of the Ionis program, because what it did is it showed us what the rates of the disease development look like, how fast the brain changes, how fast cognition changes, where in the disease we see these various symptoms and that sort of thing. And without that observational data and all of those 360 people who put in their time and effort, year after year for about 7 years, we wouldn't be where we are today. And we still have a lot more of this work to do in terms of observational trials and or observational studies, and it's really key to the continued progression of the development of a treatment. Okay, so now I want to tell you a little bit about how this drug strategy works. And Ionis is not the only company that has a drug like this, and I'll talk about that a little bit later, so I don't want you to think this is the only story in town. So the approach that Ionis uses is related to the DNA, and we know that the cause of Huntington's disease is an abnormal component of a particular piece of the DNA. And what that does, that component translates to creating an abnormal protein. So I'll tell you a little bit about that now. This is our public enemy number one. This is that protein. It's a kind of beautiful looking picture, actually. And I'll tell you what's kind of interesting is that everybody, not just people with Huntington's disease or the Huntington's disease gene expansion, everybody has Huntington protein. And the protein can be in a normal form, or it can be in an abnormal form. And people who have Huntington's disease have a mix of the normal and the abnormal form, whereas people who don't have Huntington's disease just have the normal form, okay? So that's one of the things to keep in mind. You can't tell from looking at this whether this is a normal version or an abnormal version, but I just thought I would show it to you. We never had this picture until 2016. It's the first time I got a look at this guy. Now, this is a drawing that just tries to illustrate some things about that protein, and I know it doesn't look anything like the one from before, but it's just going to help me to explain a bit about the protein. So on the left, what you have is a normal Huntington protein, and on the right, what you have is an abnormal one, okay, that's generated from the expanded HD, the expanded repeat in the HD gene. So what happens with normal proteins is that they have a good shape, and they allow the cells to function while they serve some functions usually inside the cells. And they also, you see those little green and brown and yellow things floating around. These are additional molecules that have other jobs in cells. So in the normal Huntington protein, it helps to subserve some normal functions, and it allows other molecules to do their jobs in a cell as well. But what happens to the abnormal protein is the shape gets changed. So you can see how it looks a bit disordered. The blue is kind of all over the place. And not only does the shape change, but it often kind of traps some of the other useful molecules that are supposed to be floating around in the cell. And it makes it so that the Huntington's protein itself can't really do its job that well, and the other molecules in the cell also, might get trapped and not have access to the jobs that they need to do. So this is how Huntington's disease seems to cause its havoc. And the interference with all of those cellular processes leads to changes in the brain, leads to cell death in the brain, so that the neurons that have these proteins, which are all of them, begin to get a bit sick, and then this is how you get some loss of brain tissue, and that translates to the clinical symptoms like cognitive change and that sort of thing. So what we want to have is a scenario where we have normal protein, a normal Huntington protein, but less of the abnormal one. So what's the Ionis strategy? Well, it's pretty interesting how this works, so I'm going to tell you a little bit about it. So this is called an antisense strategy. Okay, it kind of sounds like it doesn't make sense, but I'll tell you what it means. So DNA itself is important for making proteins, but it doesn't make the proteins itself. What it does is let's see if I can do a little bit of pointing on here. What it does is it creates this thing called a message. It's the messenger RNA. And the messenger RNA has the code to make the protein. And so you can see how the DNA is kind of like a ladder structure. It's like a twisted ladder. And this is kind of like half the ladder. And so the way proteins can get constructed is they can get constructed because they bind onto the matching parts of the other half of the ladder. Now what happens with the antisense drug is that they inject this drug and it gets onto the ladder and prevents the abnormal protein or the normal protein in either case from being made. So that means that this messenger mRNA doesn't get translated to make these proteins, okay? So traditional drugs, what they tend to do is they tend to bind onto the protein and try and make the protein inactive. But what this approach does is it tries to interfere with the production of the proteins overall, okay? That's the strategy. Interestingly, it doesn't just interfere with the Huntington protein being made, both forms of the protein are interfered with. And so one of the questions people had when they came up with this strategy is, if you interfere with normal Huntington protein being made, do people get worse? That's why the safety study is really important. And so now we're learning some things about that, and it looks like you can knock down some of the normal protein when you knock down the abnormal protein, and people seem to be okay. But we don't know how much of that normal protein we can knock down before it creates problems. I just wanted to put some faces to this job. So this is Frank Bennett. This is Ann Smith. Frank heads up this Ionis company and has really been the muscle behind making this happen. And Ann is the person who's developed this clinical trial and made sure that it has run really well. This guy's Ken Servin. He's a blogger who is really active in Huntington's disease. He's called Gene Veritas online if you ever want to look him up. But these are the people who've been working really hard on this strategy for years, and I just wanted to kind of show you who they were. Okay, so this is the thing. We've got phase 1/2A trial for the gene silencing drug exceeding the expectations because it safely reduces, it says here, the mutant Huntington in the CSF. I think they should have just said Huntington in the CSF because it's both the mutant and the non-mutant. But I just wanted to point out that they've got evidence so far for safety, and they've gotten the reduction of the protein. So the amount is really significant, and we know that we have the clinical finding as well. Okay, now I think I'm going to go past this, except just to say that these are like sort of stretched out images of what the protein looks like, and it just shows normally this part codes for the Huntington protein in the normal gene, and you get this elongated piece here that is because of this increased number of the CAG repeats, which we've all heard about, in the mutated protein. Okay, it's hard for me to get the right direction going there. I'm going the wrong way again. Okay, now I seem to be going the right way. Okay, so in comes Roche. So here I am to save the day, that's what they said. We are going to provide, the money that's necessary to develop this drug. This is some of the Roche team. Lauren Bork, she's actually from around country Victoria in Australia. So they're kind of an international team, and this guy's Scott Schobel. And they're in charge of the team that's working with Ann Smith and Frank Bennett from Ionis and putting the new trial together. So Roche has jumped in, and it's happening now. Okay. Now, what I want to say here is Huntington's disease affects movements, thinking, and emotions. It's not a simple disease. It's affecting a lot of different aspects of our function. And so the essential point is that we need to know, what we know so far from people with Huntington's disease is they say, yeah, the movements are a problem, but they're not our biggest problem. The biggest problem is the change in our behavior and the change in cognition, and their family members attest to that as well. Everybody's going to have their own opinion about that, but overall that seems to be the opinion that's coming through in the patient studies when they're talking about what's problematic to them. So it's not good enough for a treatment to just improve the movements. Okay, we've got to have things that improve these other aspects of the disease as well. And what's important so far with this Roche program is it does look like it has some early evidence that it might be improving cognition on a couple of different measures. We need to see whether this is borne out in a bigger study, but it looks promising. So what this press release said at the end of it was, Roche is working to quickly advance the Ionis HTT-RX, which they've now renamed because they always have a name that goes with the company. They've named it RG6042 to a pivotal study. Okay, what's a pivotal study? Well, a pivotal study is the kind of study that has enough people in it that you can really truly test the safety, and you can really truly test whether it improves the symptoms of the disease. So a study of 46 people would never be enough, because if you know a few people with Huntington's disease, what you're going to know is they're all very different from each other. It's a long disease. Some people have it when they're young, some people get it when they're older, and it looks a little different in every person, even people from the same family. So we need to have a big sample. Probably this sample is going to be 500 or 600 people in order for us to see whether the signal is good in terms of safety, and whether the signal is good in terms of improving symptoms. So that's what a pivotal study is, and if a pivotal study is successful, what you do is you take the data from that pivotal study and you go and see the TGA in Australia and the FDA in the US and the European Medicines Agency in Europe, and you have to submit this big package of data. It's about this thick on paper. You submit it and it gets reviewed in detail by scientists in each of the regulatory agencies that approve drugs. They have to review it, and then they'll give a ruling. Do they believe that the evidence is sufficient to approve the drug so that it can go onto market? And only once that happens does it become available to people, okay? There's no way to shortcut the process. That's how it works. So this pivotal study is going to be very important. It'll probably take about 3 years or so to run. It'll probably start late this year or early next year, and it'll be running in about 60 sites, probably around 10 to 15 countries around the world. So, a lot of people here might be saying, well, I want to be in the Roche study, but I want to tell you, maybe you do. And we don't know if it'll be here or not. I think possibly it won't be in Brisbane, but it might come to Australia. But we'll be trying to think about how we can get some action going in Brisbane as well as other parts of Australia and New Zealand, and make sure that our region has a good opportunity for the participation in clinical trials. We think this is really important. But it's not necessarily the best idea to be in the first trial, right? So there are other trials that will be coming up behind, and this isn't a company that's coming straight up behind Roche. They're called Wave Life Sciences. This guy's called Michael Panzera, and he works with another person that I know called Serena Hung, and they have 2 early phase clinical trials going on, just very small trials to check the safety of their drug. And they are now talking to a company. There's a big company called Takeda, big pharmaceutical company in Japan, and they've put up their hand and said, if your drug looks good in phase 1/2, we will be there to help you develop it to the next level. So these guys are coming up next. We don't know if their drugs are going to look good in the early phase trials, But some people think that it's going to look better than Roche, because the drug that Roche has, it seems to be targeting this part of the brain, but Huntington's disease, its earliest effects are deep in the brain in the subcortical structures, and some people think that this drug might be better for that, or these drugs. There's two kind of that they're looking at right now, okay? So WAVE is kind of coming up next. And there's other drug, it's the same kind of strategy as the Ionis drug, but there are other drugs coming up behind that. I heard of about a dozen of them when I was at a meeting last year. But not only that, we have other trials that are going on that are showing promising results. This trial is called the Signal trial. The company is called Vaccinex. That's Maurice Zotterer, who is an academic turned drug developer. And they have a drug that it deals with takes another approach, because what happens in Huntington's disease is the abnormal protein kind of has two major effects. It has a lot of effects, but the two major ones are it makes cells not function well, but it also causes the immune system to react in ways that can create its own problems. And this drug is aimed at trying to dampen down the immune system reaction. So it could be the way we're going to treat Huntington's disease by the time we work all of this out. We might need both, because we're never going to probably fully knock down all of the Huntington's mutant protein. We'll probably still have some of it. We'll probably still have some issues to deal with the immune system. And it might be by creating a little cocktail of these types of treatments. This is kind of how it worked in HIV. We might be able to have the best success. So that's one treatment. This is another one that's being trialed right now. This is more aimed at the symptoms of Huntington's disease. That's Ralph Reilman. He's a neurologist who specializes in carefully measuring the movements. And Teva Pharmaceuticals is a huge pharmaceutical company that just finished one trial in HD, the Pride HD trial, which kind of looked interesting, but it didn't warrant further research at this point. It's probably going to get some eventually. And then this other trial called Legato HD. So there's a lot going on. There's probably about a dozen trials going on overall looking at different drugs, but these are some of the ones that are a little bit more well developed. So that's to give you a little bit of a picture of the drug development process at the moment and what's happening kind of with industry-based drugs. There are also drugs and nutraceutical, like vitamin-type treatments that are being trialed. And I think we're going to see some of that here in Australia coming up pretty soon, clinical trials like that, and potentially those will be here in Melbourne. People are also looking at exercise and cognitive training and all sorts of things to try and slow the progression of the disease, and those treatments also look really good. I'm sorry to tell you they don't get very much funding because they're not funded by big pharmaceutical companies. They're funded by things like the NHMRC and other government research agencies, but I want to tell you that they're very promising. And I think potentially what we're going to end up with is some kind of combination of all of these things. So just to finish up, I just want to show you a couple of pictures of some of our heroes and in Huntington's disease, both research and advocacy, and then I'm going to stop. This is Nancy Wexler. She's famous for making it possible to discover the HD gene, and that's her holding a child in Venezuela where they studied huge kindreds, huge families of people with HD in the family and identified in 1993 the exact gene for the disease. This is Jim Ducella, who worked with her and was the molecular geneticist who figured it out. And he led the paper that had about 40 or 50 authors that told the community about this finding. And Ira Schultzen, who developed the best quantitative clinical measurements that have made it possible for us to start clinical trials. Because when you measure people's motor function and their cognitive function and so on, it has to all be done in a very standardized way, and he's promoted that and led a whole development of the community of clinical researchers. And Bernhard Landwehrmeyer, who led the development of the clinical researchers in Europe. And this guy and this guy are the guys who do HD Buzz. How many people have seen HD Buzz? Great, so that's Ed and that's Jeff. Ed is a neurologist and clever guy, and Jeff is a researcher and clever guy. He's from an HD family, and they're really committed to this, and they do a great job with HD Buzz. And Sarah Tabrizi, who I told you about before, she is a dynamo, let me tell you, watch out for that one. If you ever meet her, you'll never forget it. And this is Charles Sabine on the left. He's a spokesperson. He's an NBC reporter, very famous guy, and his brother who has Huntington's disease. And Charles also has the gene for Huntington's disease. He's a big advocate going all around making sure that the world finds out about HD, including the Pope, who he visited last year at this time with 120 people with Huntington's disease and their family members. And then just in Australia, we have Tony Hannon, who does animal model research in Huntington's disease. And I'm not profiling anybody from Queensland today, although that's a very good idea too. There are some good people you'll hear from today as well. Tony, this is Clement Loy from Sydney. He is getting ready to run a clinical trial on a nutraceutical. So you'll probably hear something about that. He's a neurologist as well. And Peter Panageris in Perth, he's described Huntington's disease in Aboriginal people, as well as a large group of people in Perth. Liz McCusker, who is a senior figure in the field, has been around for a long time. In fact, the person I don't have here is Ed Chu, who Jerry mentioned, he's really an important figure as well. My colleague Nellie Giorgio Christianos, who's done a huge amount of imaging work, she's based at Monash. And this is Richard Fall from Auckland, New Zealand, who's really helped us to learn a lot about Huntington's disease by studying brain tissue donated from people who have had Huntington's disease. So that's the end of my talk, and I want to thank you for your attention, and I'll be around all day, very happy to take questions. One of the things I didn't talk about is enroll, so maybe we'll have some questions about that later, but I think I better move off the stage before they get one of those hooks and pull me off. So thanks a lot for your attention.